Good day to esteemed judge, panel members, lecturer, and fellow peers. My name is Lee Zhi Xuan for the Department of Bioscience, Faculty of Science, Universiti Teknologi Malaysia. I am truly honored and excited to present my final year undergrad project at the IURC 2025 conference. The title of my project is Isolation and Antimicrobial Activity of Probiotic Strain from Fresh Cow Milk for Caviar Prevention, and it was conducted under the supervision of Dr Nurriza. First, let us start with introduction. Dental caries is a complex and progressive disease caused by caviar-gene bacteria primary Streptococcus mutant, which will demineralizing tooth structure and leads to formation of dental plaque or biofilm. According to World Health Organization, dental caries affect approximately 30% of the global population in 2019. Conventional treatment involve antibiotics, antimicrobial agents, but these often cause adverse effects such as gastrointestinal discomfort and contribute to antimicrobial resistance. Probiotics have emerged as a safer natural alternative. They're able to suppress Streptococcus mutant by producing bacteriocin, buffering the local pH, inhibiting the biofilm formation, and down-regulating the virulent gene expression of the Streptococcus mutant. In this study, I aim to isolate the potential probiotic bacteria strains from cow roast milk, a nutrient-rich untreated fluid-containing native microbial flora. While many studies have explored that probiotic from kefir or kimchi, there is limited evidence of probiotic from raw cow's milk that specifically target Streptococcus mutant. Next, we go through the experimental flow. The experiment follows a structural workflow as shown here. Raw milk samples were collected from shunt cow milk store in Kanga, Pulai, Johor. Isolation of bacterial strain was done through the severe dilution and plating on nutrient agar NA and also Denan Megosa sharp agar MRS alga. Probiotic characterization including gram staining, colony morphology, and hemolytic activity are also be done. Antimicrobial activity was initially screened using the cross-streak assay against Streptococcus mutant. Only the non-pathogenic strain that showing inhibition were subjected to further analysis, which were autoaggregation, co-aggregation with Streptococcus mutant, and cell surface hydrophobicity of S mutant, and also the EPS formation rate of Streptococcus mutant. Finally, the most effective isolate was then identified using cysteine sRNA gene sequencing. Next, we go through the most important part of my project, which is the result and discussion. So, as you see here, figure 1 shows the isolate grown on the nutrient agar and also MRS agar. A total of 8 strains were obtained which are 6 from nutrient agar where denoted as ZX1 to ZX6 and 2 from MRS agar where denoted as ZXA and ZXB. So there are 5 isolates were gram-positive bacteria which are ZX1, 4, 5A and B, while the other 3, ZX2, 3 and 6 are gram-negative bacteria. Neutral agar supported broader microbial growth, while MRS agar selectively inhibit the gram-negative bacteria and also fungi. So due to the presence of the twin anti-sodium acetate and amino citrate that can suppress the growth of gram-negative bacteria. For the probiotic characteristic, Figure 2a shows the hemolytic result. So there are only 3 strains, ZX4, 5 and B exhibited gamma hemolysis, which is non-pathogenic, and they were selected for further testing. Figure 2b represents the early screening of the antibacterial activity, which is the cross-streak assay. So all isolates show overlapping growth inhibition with Streptococcus mutant, indicating the antimicrobial substance, possibly bacteriocin. So as mentioned, only the non-pathogenic isolate with antimicrobial activity will be selected for further analysis. Hence, based on the table 1, only ZX4, 5 and B were selected for further antimicrobial assay. Figure 3 shows the graph of 4 types of antibacterial activity assay against Streptococcus mutant. First, autoaggregation that's shown in Figure 3a reflect the ability of the probiotic isolated to adhere to themselves and form predictive microcolonies. As 3 strains shown in Figure 3a, high aggregation above 95%, with ZX5 recording the highest. High auto-aggregation will allow the probiotic to form robust communicate on tooth and mucosal surface, causing a physical barrier against pathogens like Streptococcus mutans and enhancing their persistence in the oral cavity. This strong adhesion is crucial as it allows the probiotic to colonize the mouth to form a stable microcolony on the dental surface, which are easily washed away by saliva. Figure 2b shows the co-aggregation rate between the isolates and Streptococcus mutant. Based on the figure shown, ZX4 demonstrated the highest co-aggregation rate at 85.44%, significantly higher than the rates reported for Lactobacillus species in other studies. This means that this strain can physically bind with Streptococcus mutant, displacing it from the tooth surface, disrupting the biofilm initiation, resulting in no dental plaque formation. Next essay is about the formation of extracellular polysaccharide (EPS) that are produced from the Streptococcus mutant that play a major role in biofilm formation. Figure 3c shows the EPS formation rate of the Streptococcus mutant when treated with cell-free supernatant of the probiotic. It shows that ZX4 showing the highest reduction at about 83%. So, in dental caries mechanism, Streptococcus mutant are able to secrete the enzyme called glucosyl transferase that can alkalize the glucose to form glucan, which is a major product in EPS. This suggests that EP4 is able to secrete the metabolite that able to downregulate the glucosyl transferase enzyme activity, limiting the conversion of the sucrose into the sticky glucan, thus impeding the plaque formation. Finally, we go through the last antimicrobial assay. It has been demonstrated that Streptococcus mutant poses a high hydrophobicity cell surface, which will contribute to its ability to adhere on the tooth surface in the oral cavity. So, figure 3D shows the cell surface hydrophobicity rate of Streptococcus mutant when treated with CFS or probiotic, and it recorded that ZX4 has the highest reduction by 64.46%. The reduction suggests the presence of the antibacterial agent or bactericide in the probiotic that can weaken the Streptococcus mutant adherence on the teeth surface or even weaken the surface characteristic of the Streptococcus mutant. To summarize, ZX4 consistently outperformed ZX5 and ZXB in all parameters, including high auto-aggregation rate at 95.61%, highest auto-aggregation rate with Streptococcus mutant at 85.44%, strong reduction in EPS production and hydrophobicity of Streptococcus mutant when treated with cell-free supernatant of ZX4. Thus, only ZX4 will be selected for molecular identification. Genomic DNA of ZX4 was extracted using the World Genomic DNA Verification Kit. The 16S rRNA was amplified using universal primer called 27F and also 1492 reverse primer, followed by 16S rRNA sequencing and phylogenetic analysis using MegaChart software. So, the phylogenetic tree shown in figure 4 suggests that ZX4 shares 83% similarity with Bacillus licheniformis 00147104. This bacteria has been previously described as a probiotic species with the antimicrobial properties, gut microbiota modulation potential, and also other health benefits by other studies. In conclusion, this study successfully achieved the objective by isolating the probiotic strain from raw cow's milk with ZX4 Bacillus licheniformis, demonstrating the most promising antibacterial activity against Streptococcus mutans. However, further studies are needed to confirm its safety and also determination of the minimal inhibitory concentration value, biofilm formation, and also the antibacterial activity using agar-well diffusion assays. And finally, investigation of the species ZX5 and ZXB. Finally, I would like to extend my deep gratitude to Universiti Teknologi Malaysia for providing me a platform to participate in IURC 2025 to share my research findings. Thank you very much for your time and attention to listen my presentation.